we call these raised areas, what I would describe as little hills, we call those a gyrus for a single, or gyri for the plural. We call the little spaces down in between, which I can't really get into because my pia mater is over there, but if I cut through my pia mater, I can really get down in there and do a blunt probe. You can see that there are actually grooves. We learned this back when we learned bones. They're called a sulcus for a singular, or sulci for the plural. Some people say sulci, some people say gyrus instead of gyrus. Either one is fine. We have some bigger grooves on the brain surface. The major groove that divides this longitudinally is called the longitudinal fissure. We're going to cut through that here shortly so we can take a look at things. Sometimes it is described as the commisural fissure, C-O-M-M-I-S-U-R-A-L, because it is the crack just above the bridge between the right and the left hemispheres, and a commissure is a bridge or connection of white matter tracts or fibers that connect across the brain. So this goes down and stops right where we have the crossroads that go through there, the commissures. So then we have a couple of sulci, not as visible on the sheep brain. On the human brain, between the frontal area and the parietal lobes, frontal lobes and parietal lobes of the brain, you'll see a groove that goes across like the same as a coronal or frontal suture, and that's called the central sulcus. And then if you flip the brain sideways, and again, you don't see this quite as well in a sheep as you do in a human brain, but there's a groove right here between the temporal lobe and the rest, and that's called the lateral sulcus. So theirs is kind of buried down in here a little bit. That lateral sulcus opens up into an inner fold in the human brain called the insula. It's just like it's an inner layer of more of these gyri. If we look at the brain from a lateral view, let's see if we can get that lined up pretty well, you'll notice there's sort of a smaller brain and a larger brain. Now in the sheep, that larger brain isn't a whole lot larger, but in a human being it would be much more pronounced, and then this smaller brain would be quite a bit smaller. Our general rule with Latin is, the longer the name, the tinier the structure. So the cerebellum is this small brain area. The cerebrum, which is the shorter name, is the much bigger, more what we call the higher brain. This is where we have higher order functions going on. We call most of these, this area where there's these folds and grooves, we call it the cortex. The cortex kind of grows up like a stem, comes up like a flower, and the petals fold down around that brain stem again. So this part right here, this brain stem in here, which we'll take a look at when we cut this in half, we do sagittal section, this brain stem is referred to, the lowest part is referred to as the medulla oblongata. It's the medulla because the cortex, it's in the middle, and the cortex grows down around it, covering it. Many other structures in the body have a medulla and a cortex. Kidneys, lymph glands, adrenal glands. So this is the first time in our experience in AMP1 that we've really talked about a cortex and a medulla. It's not quite as accurate in terms of being in the middle and then surrounded by a layer, but you kind of see it. So if we flip the brain over and we look at the inferior side, what we can see, I'm going to put this aside just for a moment, what we can see coming out the front, very, very well developed olfactory nerves on sheep, very dependent on their sense of smell. They have a better olfactory tract than we do, more developed. Their optic nerves are quite well developed. They have good vision. And what happens, what you'll notice with optic nerves is that they have an area where they cross. It makes an X, which we refer to as the optic chiasma. The letter X in the Greek alphabet is referred to as chi, or chi. Some people say chi. I learned it as the chiasma. So you see this X marks the spot, and that brings information from this right eye back to the right side of the brain and also to the left side of the brain. So it can crisscross both of those, and that helps with not only 3D vision but general visual interpretation. Now, if I kind of pull that aside, then we move down the pike here a little bit, you can see more of the stem structures. We're going to cut into it now so we can really see on the inside. So now we're going to do a sagittal section. We're going to start at the medulla down here, which actually looks kind of like brain stem. My hand is blocking the view here. Sorry, folks. There we go. I'm going to go right up the middle of the cerebellum. I'm going to go right up the middle of the longitudinal fissure or commisural fissure. And we'll have a lovely sagittal section, similar to what you'll see in the human anatomy textbook. Okay. We see, when we look at this lateral view, sagittal view, we see the medulla is kind of this widened place of the spinal cord. Then right across, the next kind of major structures up, we see the pons, which is a little bit larger in the human. It's an oval, just a small oval here. And then the cerebellum is directly across from that. They're neighbors. I like to think of the cerebellum as the mansion with all the rooms where you store information. And then it looks out over the pons. So it looks down its lawn and across the lawn to the pons, and then you can remember they're right next to each other. The next section moving up the brainstem, and in terms of development, the next section that develops in the brainstem is the midbrain. And this is involved with relaying sensory information, and it's particularly involved with hearing and visual reflexes. Then we're going to move up, and you'll see kind of a round or oval. It'll be a little bit darker structure up at the top of this brainstem called the thalamus. And that's the final processing relay station. All information coming from the body goes through these structures and ultimately through the thalamus and is relayed to which part of the brain it needs to go. Does it need to go to the visual center in the occipital? Does it need to go to the hearing auditory center in the temporal area? Does it need to go directly up here to the primary sensory, somatic sensory area in the post-central gyrus of the cerebrum up here? So, and then atop that, you'll see a space. If we kind of separate this, you see some spaces in here. These spaces are referred to as ventricles. Just like in the heart, where you may have heard ventricles used before, these are chambers. They're filled with cerebrospinal fluid. In this chamber here, there should be a vascular structure. You'll see some blood vessels down in here that look dark. That's the choroid plexus, where the cerebrospinal fluid is strained out of the plasma from the blood supply, circulated through the central nervous system, back up around the brain, and then out into the venous, through arachnoid granulations up here, into the venous circulation, and back into the body again. If that gets blocked anywhere along the way, hydrocephalus can result, where the fluid builds up and alters the size of the cranium. It stretches the cranium out as it's developing. If you don't relieve that pressure in there at some point, it could be fatal. So we have a ventricle here. We have 4 ventricles. There are 2 lateral ones that sit under this corpus callosum here, this kind of white-colored C structure. And then there's a 3rd ventricle that comes down this little space in here just above the cerebellum. And then there's a 4th one right in here where the cerebellum is right next to the spinal cord, across from the pons. So the next thing we want to take a look at is this corpus callosum is white matter. Anytime we see white matter in the central nervous system, and anywhere, it's myelinated, which means it has some kind of a glial cell. That would be a neurolemmacyte in the peripheral nervous system and an oligodendrocyte in the central nervous system that makes a lovely fatty coating to protect those axons so information can travel more quickly and more efficiently from gray matter to gray matter. So the gray or tannish color matter, it's called gray matter because in freshly harvested specimens or in a living brain, it is actually gray. Once we put preservative on it, it turns a little bit more tannish. But the darker areas are gray. That's dendrites and cell bodies and synaptic terminals. And the white matter is myelinated axons. So you see this is information moving, this is information connecting. The gray is where the connections happen. You'll notice sheep send a lot of information, but they just don't process very much. If anybody's ever worked with sheep, they're pretty stupid. They do not train well, they do not handle well, and they live mostly in their very well-developed brainstem, which keeps them on animal instincts most of the time. We, on the other hand, have a lot more cerebral cortex up here, which I believe we have evolved to help us override the biological urges in this part because we have so much more up here, but that's for another discussion. At any rate, you can really see this white and this gray matter pretty well once you slice into it. I think we've covered major stuff, most of our stuff. On some of the brains attached on this dura, you will actually see there's a pituitary gland and a mammillary body may be stuck on there. So you may want to take that dura off of there and feel it'll be very spongy. Glandular tissue is very sponge-like. And so you can kind of take a look at that. Other than that, I think you may want to cut the brain in a coronal section or frontal section and then take a look internal. This way you can see a ventricle right here is a lovely ventricle. So when you're looking at it in this direction, and you can see that. And actually, if I put those together, let's see if we can see where a commissure goes across there. Okay, here is the corpus callosum, the white that connected across. Here's a ventricle on each side, the two lateral ventricles, and here is the longitudinal or commisural fissure that comes right down to the bridge. And if I hadn't cut this, this would have been solid white right across here. Okay. Also, areas where you find a lot of gray matter kind of in an island like this, we describe those as a nucleus in the brain. And I think that's all. I think that's about the best we can do with a sheep brain. Study hard.